Radiopharmaceuticals are diagnostic and therapeutic agents. So they consist of a targeting agent, so typically a small molecule or an antibody, and this will target the tumor. There's also a radionuclide, and there's a linker that connects the two. And the radionuclide can act as either an imaging agent for diagnosis of cancer or as a therapeutic agent, where the radionuclide will deliver a lethal dose of radiation to the tumor.
What if the next breakthrough in cancer treatment isn't a pill or an infusion, but a precisely targeted radioactive payload that hunts down tumor cells like a guided missile? Today, we are diving into the radiopharmaceutical revolution with Bryan Miller, who is the Director of Scientific and Technical Operations at Crown Bioscience UK. From his early days studying colorectal cancer models to now leading preclinical development for one of oncology's hottest therapeutic classes, Bryan is taking us inside the science that's transforming how we fight cancer. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Bryan, to the Smart Biotech Scientist podcast. It's good to have you on today.
It's great to be here, David.
Bryan, share something that you believe about early discovery research that most people disagree with.
So I've found that research progress is rarely linear or predictable. Over my career, I've seen the undruggable become druggable. I've seen false starts in some areas and surprisingly rapid advances in others. And I think that's why I find our field so dynamic and exciting and why I love being a part of it.
Excellent. Draw us into your story. Tell us what sparked your interest on working on cancer models, and then what were some interesting pit stops along the way that got you to your current role?
Yeah, it's quite interesting. So my PhD was actually in cardiac disease. So that's where I started out. And it was when I did postdoctoral fellowships, first at the University of Toronto and the second at CRUK. That's really what took me towards oncology models, both in vitro and in vivo models. And my postdocs were focused on models of colorectal and pancreatic cancer. And I think what motivated me at that point is exactly the same as what motivates me now. I was passionate about contributing to the development of new therapies for cancer and identifying new therapeutic targets. And really, that's what motivates me to go to work every day to this day. Being part of Crown allows me to work across a large array of different cancer indications with a diverse range of clients. And it's really motivating when you're contributing to the development of new therapies. And it's fantastic when you've contributed to a research program that has led to a therapy going into the clinic.
Tell us a bit more about your current work. How does that look like when you're developing these new therapies?
So we're a preclinical contract research organization. So we're oncology focused, but we do work with a large array of different clients working on various different therapeutic types. And we have a range of advanced preclinical models that are available to our clients to run the research programs with. So we do work across most cancer indications and we do work across with most therapeutic types. So it's a very diverse array of work that we do conduct at Crown, which makes it really interesting for us. It's very varied, it's very interesting, and it's absolutely fantastic to support this variety of client research projects.
And when you say it's very diverse, so that includes the various diseases you work on, I imagine also very diverse companies, which have very diverse needs. Before we dive a bit further into the radiopharmaceuticals, what is the main need companies have that come to you? What is the main problem they want to solve?
So they typically come to us, usually with a target in mind and usually with a therapeutic that they wish to test. And really, one of our main roles is to guide them towards the most appropriate preclinical model to address the question that they're asking. And we do have a range of different models available. We've got an excellent array of in vitro models, particularly organoids. We're well known, I think, for our organoid platform. So certainly if you get the in vitro phase, we do have an array of advanced in vitro models that can be very, very valuable. We're also very well known for our PDX library. We have PDX models that cover most cancer indications. And we have genomic data from them. If you have targets in mind, we can guide you towards the most appropriate PDX model for your work. In addition to that, we have an array of CDX and genetic models, humanized models. So really, I think we're cancer focused, but I think for most questions around development of cancer treatments, new oncology therapies, we will have appropriate models for you to use in your research program. And we can help you filter those down and decide the most appropriate ones for you.
When you say models, are these animal models or are these 2D cultures, 3D cultures, or is it a combination of various models?
All of that, actually. So yeah, I've got 2D cell culture, 3D cell lines, organoids. We've got various in vivo models, so syngeneic models, CDX models, PDX models, humanized models. So it really is quite a wide array.
Excellent. So let's dive into one specific area, the radiopharmaceuticals. A lot of our listeners, I imagine, are not familiar with that. So can you start with the basics? What are radiopharmaceuticals and how do they differ from traditional cancer therapies?
Yeah, so radiopharmaceuticals are diagnostic and therapeutic agents. So they consist of a targeting agent, so typically a small molecule or an antibody, and this will target the tumor. There's also a radionuclide, and there's a linker that connects the two. And the radionuclide can act as either an imaging agent for diagnosis of cancer or as a therapeutic agent, where the radionuclide will deliver a lethal dose of radiation to the tumor.
Can you just elaborate a bit more? Give us an example. How are they different from a traditional cancer? Is this the morphology? Is it the way, or what is it exactly? What are the main differences?
I'll say there's probably two real advantages of radiopharmaceuticals. First is safety, because you're dealing with a therapeutic that can very specifically target a tumor. So one of the early stages that you do is you characterize the level of accumulation within the tumor and make sure you don't have accumulation in other organs of the body. So they can have a greater safety profile than more traditional therapies. That's one advantage. The other thing that distinguishes them is the idea of theranostics. So you can use an agent that combines diagnostics with therapy. So you use the same scaffold. You can use one radionuclide, which will allow you to diagnose a tumor, and potentially switch it to a second radionuclide, which will allow you to target the tumor and to kill the tumor. And because you are combining those within the same strategy, this allows for, firstly, really good stratification of patients, but also secondly, very rapid progress from diagnosis through to therapy.
Okay, I see. So it's faster and it's also a lot more things, I'd say, happening in parallel to get a much broader picture.
Yeah, and I think safer. I think a lot of them are much safer than traditional chemotherapies.
So the radiopharmaceutical field has gone from a niche therapy to now one of the hottest places in oncology. Tell us, what is driving this rapid transformation in both the science and also the commercial interest? I mean, there's a lot, I imagine, happening on the scientific side, but then also the commercial interest, investor side of things, right?
I mean, that's what we're finding as well. I think seems to be growing interest in the area. It's quite a dynamic area right now. And I think there's a lot of exciting things happening in it. I think it does go back to some of those advantages that I just described. The theranostic approach, because that really does give these therapies quite a considerable advantage. I think the safety profile is another thing that a lot of companies we are working with are finding very valuable in the development of the therapies. And I think maybe the third aspect is just widening the range of cancer indications that radiopharmaceuticals are available for. So we are starting to see, more radiopharmaceuticals coming through that are starting to target a more diverse range of cancers.
And the main purpose of all that is basically to make faster, actionable, and more predictive decisions with all your models. I'm just curious, on top of your models, do you add a layer of machine learning, AI, advanced data-driven approaches, or how does that work in your field?
In terms of model selection?
Yeah, and also in terms of interpretation of the data.
I would say that is certainly applicable to the selection of models. I mean, absolutely crucial. You need to get your preclinical model right in order to have a successful research program. And we do have large data sets associated with all of our models. So certainly if you're looking for particular targets, particular gene profiles, then we can help you with that. We have these models very, very well characterized, and it's certainly information that we're happy to share with you in order to guide you towards the most appropriate model for you. We're also very excited by the prospect of AI. I think that is something that is much more coming online, and I think it is something we will be incorporating into workflows as well.
Yeah, you're making a very good point that you have to select the right clinical model because that's the foundation, isn't it?
Exactly, yes. If you get the model wrong, you're not going to have a successful program.
Yes, you got to solve or answer the wrong question at the end of the day if you choose the wrong model. Besides that, what are other pitfalls when you're developing a new drug? What other choices or I'd say strategies have to be right?
Yeah, I mean, particularly in the radiopharmaceutical field, it can be quite a challenging area. So I think an early point is you need to get the targeting strategy for the tumor correct. So I think that's a very important aspect. You need to be able to target that tumor and have a lot of specificity in that targeting as well. You also need to select an appropriate isotope and a correct labeling position and labeling strategy. Another important aspect is you need to make sure either you have or you're working with someone who has a reliable source of radionuclides. These materials do have to be labeled fresh before dosing, so you have to make sure that supply of radionuclides is reliable. And really, really crucial is to have a robust labeling strategy and to make sure that proper and thorough QC is done on the materials that are labeled. And nothing should be dosed unless it passes quite stringent QC tests. As we discussed, make sure that the actual model you're selecting is the most appropriate one and is the one that's going to give you the best chance of success. And those are all areas that we can assist you with. We can take you through those areas, we can give you advice, and we could help guide you in designing the most appropriate study design.
And compared to, I'd say, conventional drug development, what are the unique challenges that related to the radiopharmaceutical development? Because you mentioned now quite a lot of things that have to be right. How different is radiopharmaceutical development compared to conventional drug development? And what are the unique challenges?
I mean, obviously there's, some commonalities, but I think the unique challenges would be that need to have it prepared fresh. You can make it in bulk and store it. You have to have fresh material that can pretty much be dosed right away. And then also the complexity of some of the strategies, designing an appropriate labeling strategy. Because it's quite a specialized area, you really do need to work with people who have specialist understanding of it and can essentially advise on the most appropriate way to label it, the correct isotope to use, and also crucially the linker to use. I think those are all aspects of the design of a radiopharmaceutical that are going to be absolutely crucial in order for it to be successful.
And how do you in your company manage all that and navigate these difficulties? What are some specific approaches you have to develop?
I think absolutely crucial for this is the collaboration that we have established with Medicines Discovery Catapult. We've recently launched this strategic collaboration with MDC. And what this does is it brings together the range of preclinical models and preclinical expertise from Crown Bioscience with the radiolabeling expertise and the experience with radiopharmaceuticals from MDC. And it's a really, really strong partnership. And I think we're combining real strengths and real complementary experiences to deliver highly successful radiopharmaceutical studies for our clients.
And what kind of clients do you usually work with? What kind of companies are they? Are they small companies, mid-sized, or even the big ones, the big pharma companies?
Yeah, all three actually. We've got a diverse range of clients, small, medium, and large. And as we were alluding to before, there's a growing interest in the radiopharmaceutical field. So we are finding more and more people approaching us to discuss radiopharmaceuticals and discuss appropriate strategies for the further studies. And that does range from quite small companies to larger ones.
I imagine their needs are quite diverse. Can you just give us a highlight or a preview of how much the needs differ between the small and the big pharma company? What are the typical needs they have and the differences between them?
From what we've experienced, I would say one of the differences in how mature the program is. We're finding that some people approach us with really at a very, very early stage. So they won't have a strategy for labeling the test material yet. So in that case, we may have to do a lot of the optimization. We may have to guide them on isotope selection and perform a lot of the validation work to make sure that you can label the material properly and that it's suitable for use. We find some other clients, their program will be a little bit more mature. So they will have optimized the labeling already. They will come to us with a protocol. We may perform the labeling, but it is from a protocol transfer rather than setting it up from scratch. So I think those are two quite different requirements that we've found with clients approaching us.
Since the interest is in radiopharmaceutical development is increasing, both on the scientific side and also on the business side, I imagine that you're getting more and more demand for such a development. How do you make sure you can scale your preclinical and translational capabilities to meet the demands of all these companies coming to you?
I think we've got a lot of experience with it. Both ourselves and Medicines Discovery Catapult have worked in our respective fields for many, many years, and we are used to running a wide array of projects simultaneously. And so scalability is something we are very experienced in. But you're absolutely right. We are finding increasing demands for these types of studies. But it's exactly the same principle as we would use for other studies in terms of scalability of our in vivo models and from the NDC side, scalability of the radiolabeling side of the project as well. And both parties have a lot of experience in delivering these studies and a lot of experience with scalability.
That's where we will pause for today. In part 2, we'll explore the specific platforms accelerating radiopharmaceutical translation and get Bryan's predictions on where this field is heading. If you're finding value in these conversations, please leave us a review on Apple Podcasts or your favorite platform. It helps other biotech scientists like you discover the show. See you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.